Good morning, everyone. After the invigorating sessions on energy, climate, and AI, I'm thrilled to welcome you to the Life Science session. I'm Haifan Lin from Yale University, and I have the honor to moderate this exciting session. As you all know, life science is an internal theme that has fascinated humanity since the dawn of civilization. From marveling the wonders of life to grappling with the challenges of sickness and death, humanity has relentlessly pursued the enhancement of health and longevity. Today, research in life sciences is spearheading a new revolution, transforming how we live. In this one-hour session, featuring a keynote speech and a panel discussion, we aim to capture this excitement. We are honored to have Professor Randy Schekman to deliver the keynote speech. Dr. Schekman is a professor at UC Berkeley and an investigator of the Hughes Medical Institute. His remarkable journey in science began as a PhD student with Arthur Kornberg at Stanford University, where he studied DNA replication. He then conducted postdoctoral research with Seymour Singer at UCSD, where he devoted lifelong interest in cellular membranes. At UC Berkeley, Dr. Schekman has made pioneering contributions to our understanding of vesicular traffic in the secretory pathway of eukaryotic cells. His outstanding work has earned him the highest accolades in science, including the Gairdner International Award, the Lasker Award in Basic Medical Research, and the Nobel Prize in Physiology or Medicine in 2013, among many other honors. Beyond his research, Dr. Schekman has trained numerous world-class scientists and is a prominent leader in the scientific community. On a personal note, I have had the privilege of working with Dr. Schekman on various committees and have been continuously inspired by his passion, dedication, and high principles in serving the scientific community. Please join me in welcoming Dr. Schekman.
Thank you very much, Haifan. And I'm delighted to be here today. I'm thrilled that Yi Choi invited me to come back to my alma mater. I was a graduate student here many years ago. Today, rather than discussing my own research, I wanted to share with you an initiative of international reach that provides an example of collaborative research that can overcome political and geographic boundaries. And in this instance, the influence of a sizable philanthropic donation that is not burdened by the rules and politics of government. It concerns a scourge of humanity, a disease that we've known in some detail about for a very long time. It was first described 200 years ago, Parkinson's disease. Parkinson's disease has reached pandemic proportions. It's believed to continue to rise more rapidly than any other neurodegenerative disease that mankind suffers from. In the next decade, there will be estimated maybe 40 million people who suffer from Parkinson's, 10 million in this country alone. It knows no geographic boundaries. It's estimated that in the next decade, over half of the new cases of Parkinson's disease will emerge in China, so this is of vital interest around the world. About 25% of the cases of Parkinson's that we know about have some genetic origin. There are now over 20 different genes in familial forms of the disease. And yet, the majority of cases, perhaps 70% or more, are of sporadic non-identifiable sources, possibly environmental toxins that have not yet been adequately identified. The disease afflicts people at a variety of ages. It may set in at a young age with, in this case, early dementia, rapid disability, or sometimes it can set in at an old age. Sometimes it can progress very slowly. For instance, the actor Michael J. Fox, who is a major proponent through his organization, the Fox Foundation, has suffered from Parkinson's disease for nearly 40 years. Others set in with dementia right away and don't survive. It's been treated, though, by the pharmaceutical industry as a single disease, and yet it is likely a spectrum disorder with many origins. People present with different symptoms, they progress at different rates, and they die for different reasons. It is not a single disease. We must consider it with greater granularity. Let me tell you a little bit about what we know about the morphological basis of the disease. Here is an image in 3 parts of one of the key molecules that is known from genetics and in the sporadic forms of the disease to be important in the spread of pathology in the brain. In the upper left quadrant, you see a section of the human brain, the midbrain section from 2 patients who died. One on the right, a patient who died for other reasons, has a region of the midbrain called the substantia nigra that can be visualized because it accumulates the pigment melanin. In spite of this being deep in the brain, this pigment exists because of an oxidation of a neurotransmitter dopamine of critical importance in the disease. When patients first present with the symptoms of Parkinson's disease, the typical movement disorder or tremor, it's estimated that they have already lost over half of the tens of thousands of dopaminergic neurons in this region of the brain. And by the time someone dies, such as in the instance on the left, most of the melanin pigment is gone because those cells have died. Now, on the right, about 100 years ago, a German clinician, Friedrich Louis, on inspecting sections of the brain that include these dopaminergic neurons, discovered inclusions, heavily stained regions of the inside of these nerve cells that are darkly stained. And in his honor, this structure has been given the name Lewy bodies. It is a pathologic hallmark of the disease, but unfortunately it can only be seen, at least thus far, in biopsy samples from patients who have deceased. We can't really yet evaluate these Lewy bodies in living patients to mark the progression and movement of the disease. Now, at the basis of this aggregated form of material in the cell is this protein shown pictorially below, a very small brain protein called alpha-synuclein. And patients who have mutations, 3 of which are highlighted by these arrows, produce a form of the protein that tends to aggregate, to clump together inside the cell, which may be the nucleus upon which other things within the cell accumulate to create Lewy bodies. It's also important to control the abundance of this protein because in a very rare genetic form of Parkinson's disease, this gene is copied 3 times, a genetic triplication, which results in the overproduction of this molecule, which then allows it to be more aggregation prone. Now, subsequent to the discovery of the Lewy body, another German clinician, Dr. Brock, sectioning many brain biopsy samples from patients who died from Parkinson's and for other reasons, was able to put together a speculative model about how these structures may advance within the brain. In the earliest forms of the disease that he could see, these Lewy bodies were seen in the brain stem and also in the olfactory bulb. Indeed, patients who present with Parkinson's disease very often, many years earlier, sometimes decades earlier, have a poorly developed sense of smell. This is not necessarily diagnostic of Parkinson's disease, but it is striking what fraction of these patients advance eventually, many years later, to Parkinson's. As the disease progresses, these Lewy bodies may advance to the region including the substantia nigra, and eventually, near the end of the disease, these Lewy bodies extend into the higher regions of the brain and may affect emotional and cognitive functions. Now, let me tell you about some parts within the cell that are particularly key choke points that are likely involved in the initiation and possibly the progression of the disease. The first organelle I'll draw your attention to is the kind of energy battery of a cell. It's called the mitochondrion. The mitochondrion is shown here. It's a structure with, It's a structure with a two-membrane envelope. It has an interior core of many enzymes that are involved in, some of them involved in producing the energy currency of the cell called ATP. It also has its own little genome, a little small mitochondrial genome. All of these things are essential for cell metabolism and energy production. And these mitochondria must remain vigorously active in all the cells in our body to sustain normal metabolism. Cells have evolved, and organisms have evolved, a powerful surveillance mechanism that allows the cell to tell when a mitochondrion is damaged. Because if a mitochondrion becomes damaged, sometimes the chromosome within the mitochondrion can escape into the cytoplasm of the cell. And cells don't like to have DNA in the cytoplasm, and so that elicits a death spiral that kills the cell. So cells want to protect the functioning mitochondria, and they want desperately to get rid of damaged mitochondria. There is a path to do that, which I've illustrated on the next slide, called autophagy, or basically the cell's ability to swallow up damaged material. In this case, the damage can be seen in the form of both a mitochondrion and a different organelle. Once they become damaged, they are recognized, they become enveloped by a membrane that surrounds these organelles, and they are then delivered to a different organelle in the cell called the lysosome, which is a kind of a digestive tract. Once in the lysosome, the mitochondria and other things are chewed up into little bits, and the constituent molecules are delivered back into the cytoplasm for the cell to use. Now, this is particularly relevant to Parkinson's disease because 2 of the genes of the 20 that are found in familial forms of disease affect the ability of the cell to surveil damaged mitochondria. Let me show you an example of these 2 genes and what they do. So in this image, we're looking at a normal mitochondrion that's properly healthy functioning. And if the mitochondrion is damaged, this can be done experimentally or happens in a body. 2 proteins are activated that recognize the damaged mitochondria and attach a little tag, a little string of amino acids, a peptide called ubiquitin, becomes attached to the outer surface of the mitochondrion, and that little tag is a kind of an eat me signal that elicits the formation of a membrane that surrounds the damaged mitochondrion and delivers it into the lysosome for degradation. Now, patients with mutations in these 2 genes, which are called PINK1 and Parkin, mutations in these genes prevent the cell from making this tag, recognizing damaged mitochondria. Therefore, the damaged mitochondria persist, and that wreaks havoc with the cell and causes eventually cell death. Now, in the last example I'll give, another gene that's involved, a gene that is an enzyme. It's called LRRK2, LRRK2, a very common mutation in this gene, interferes with a novel signaling pathway, that allows cells that make the neurotransmitter dopamine, that is the basis of most of the pathology of Parkinson's, to communicate with another nerve cell some distance away in a higher region of the brain. These two cells have a pathway of signaling, which has the funny name, the hedgehog signaling pathway, and they both function and sustain each other in viability and in function. The mutation in this gene, mutations in the LRRK2 gene, interrupt this pathway of communication, causing a failure in communication, which results first in the death of the dopaminergic neuron, and then in the loss of this other neuron in the higher region of the brain. So there are many different pathways that reach out and touch dopaminergic neurons to block their function. Now, this is the typical view in a textbook, a cartoon that you use to visualize what a nerve cell looks like. This is a false image. It looks nothing like what actually happens in the brain, but it illustrates the one point that I wish to make. The nerve cell has a cell body with a nucleus and mitochondria. It has dendrites facing one side of the nerve cell that serve to receive communication, and then a long axon and nerve terminals, which may engage many other nerve cells. It's estimated that there are tens of billions of nerve cells in the human brain, and each nerve cell can make usually 100 or more connections to other nerve cells. Sometimes these connections vastly exceed 100. Nonetheless, there are likely over 1 trillion different nerve cell connections in the brain, and these inform all of the processes of learning and memory and emotion and cognition that define us as human beings. Now, one particular nerve cell, the one that's implicated in Parkinson's disease, that is remarkably vulnerable, you'll see in the next slide, is the dopaminergic neuron. Here's an image of what a dopaminergic neuron looks like, very different than a normal nerve cell. It has a cell body, and it has dendrites, it has an axon, but instead of the dozens or hundreds of possible nerve connections, it's estimated that a dopaminergic neuron has 1 million or so different possible nerve connections. That's enormously complex. So you can see that this cell, though it's enormously complex and terribly important, is also particularly vulnerable. It is poised on the edge of viability. It requires all the energy that mitochondria can produce. Any diminution in the function of mitochondria in these cells will lead the cell, eventually propelling it to death, which is why even those of us here who will never have Parkinson's disease are losing dopaminergic neurons all the time. They are only rarely being replaced. So mutations in the genes that I've described that seem to be specific, they're having a specific effect on the viability of dopaminergic neurons, do so because that cell is more vulnerable than most of the other cells in our body. That's why these mutations have a very specific effect. Now, why did I become involved in Parkinson's disease? It relates only peripherally to the nature of my own basic research at Berkeley. I became involved because my wife, Nancy Walls, suffered from the disease. I met Nancy when I was a graduate student here at Berkeley, at Stanford many years ago. We were married in Huddart Park, not too far from here. We moved to Berkeley, had a wonderful life together, raised 2 lovely children. Everything was fine for the first 25 years of our marriage. And then, around the late 1990s, she developed some unusual symptoms that turned out to be because of Parkinson's disease. Nonetheless, she was perfectly normal. Here's a picture of Nancy. She was perfectly normal for the first 5 years because there's an effective therapy to replace dopamine that for a number of years can mitigate much of the symptomology of the disease. Nonetheless, these medications don't change the arc of the disease. The disease continues inexorably. And within 10 years or so, she developed a lot of the dyskinetic movements that are associated with the disease. She had a neurosurgical procedure called deep brain stimulation, which corrected many of the problems with the disease. Nonetheless, the disease advanced. She developed dementia. She became quite dependent on continuous care. Nonetheless, there were some happy moments together. Here's a picture of the banquet in Stockholm in 2013 showing a memorable picture of my Nancy and my father sitting together enjoying the event. She was able to come. I was grateful for that, but she had to be managed by my entire family to keep her healthy. In the years after that, the disease accelerated, her dementia worsened. I went to neurologists around the world and asked them what to expect. I knew there wasn't going to be a treatment, but what could I expect next? They'd give me an answer, and they were always wrong. Here she was, within months of death. She was catatonic much of the time. She was incontinent. She couldn't take her medications. There was really nothing left to do. She died in the middle of the night, likely of heart failure. No one ever warned me of that. One of the frustrating things about dealing with a family member as a caregiver is the feeling of helplessness. There really is nothing you can do other than to make them feel comfortable because the disease progresses in unpredictable ways. After she died, I was approached by the chief financial officer of the Sergey Brin Family Foundation. Knowing what I had done in science and knowing my personal situation, I was asked if I would consider helping them invest money in the basic science of Parkinson's. Mr. Brin, as you know, a Stanford grad, enormously successful, but he has a family genetic history of Parkinson's disease. He and his mother share mutations in the gene that I described called LRRK2. His mother is seriously ill, progressively with Parkinson's, Mr. Brin not yet. But he has been investing in Parkinson's research for many years now, first with the Michael J. Fox Foundation, the major philanthropic arm of Parkinson patient care in the country. But Mr. Brin decided that in the history, the 200 years of history of the disease, there was really nothing to change the path of disease. There was nothing to dampen or slow or arrest the progression of the disease. And he felt the problem was that we didn't know enough basically about the molecular and cellular origin of the disease. And so, having just lost my wife, I decided to drop other things aside from my own research that I was doing, and I joined forces with a small team of people to create a new program with a very significant investment of philanthropy from Mr. Brin that I'd like to share with you in the next few slides. So the program is called Aligning Science Across Parkinson's Disease. And during the first 2 years of our existence, my partner, Kemeny Riley, who is a PhD molecular biologist with an office in Washington, and I set up a program to identify teams of investigators, not individual, but teams of investigators who had a common interest not only in this problem, but who had already experienced the joy of real collaboration, meaningful collaboration. And so we set out applications around the world. We received hundreds of applications from teams. And let me tell you the themes of the research that are now under investigation by our collaborative research network. We, first of all, had as our goal to provide a strategy and a mechanism, and I'll tell you what that strategy is, for directing the financial and human capital to solve this critical basic science problem. We viewed Parkinson's as a puzzle, obviously more complex than this puzzle, but nonetheless with some of the same parts: dysfunction of the mitochondrion, problems at the synapse, a problem of misfolding of this protein alpha-synuclein, problems with degrading proteins, a problem associated with inflammatory process in the brain that may initiate or exacerbate lesions, and many other unknowns. We estimate thousands of puzzle pieces need to be fit together to create a picture that would provide the pharmaceutical industry with the clues that it needs to make really meaningful advances. After the 2 years of study involving collecting opinions from many neurologists and basic scientists around the world, Kemeny and I came up with these 4 pillars on which we base our investment. They are, first, the genetics and associated biology, those 20 or more genes where there are individual proteins of known function. How do they fit in together? That's an obvious target of opportunity. The next is the inflammatory process that I mentioned that is evident not only in Parkinson's but in Alzheimer's disease. Another is the circuitry, the brain circuitry. Imagine the complexity of a dopaminergic neurons innervating other nerve cells. Obviously, that has been incompletely mapped, and we want just to understand the complete circuitry if possible. And finally, and most curiously, a syndrome that often precedes Parkinson's. I mentioned one syndrome, simply a poorly developed sense of smell. But another one that proceeds often within 5 years of diagnosis is a disease called REM sleep disorder, sometimes called RBD. This is where patients live out their nightmares and flail around in bed. They've lost the ability to suppress the movements that are associated with dreams and nightmares. Pathways that operate in all of us perfectly normally are somehow disrupted in pre-Parkinson patients. And these patients often flail and throw themselves out of bed. They end up fighting in their nightmare with their partner. It can be very dangerous, very frightening. What is the connection between that and Parkinson's? We really have no idea, but within 5 years, 80% of people who have RBD advance to Parkinson's disease. Well, when we put out the call, there were other themes, more sociologic themes that we felt very strongly about that I want to share with you near the end here. The first was to identify teams, and this was crucial. We wanted teams, not individual superstars. We wanted teams, effectively integrated teams, who've developed productive, meaningful collaborations to achieve goals that supersede the expertise or capability of any one lab. We wanted to attract diverse talent, not only from within Parkinson's, but outside in allied areas of some potential benefit. We wanted to drive the focus on those 4 themes very closely. And we wanted to embrace the values of openness and transparency. We require all of our investigators to communicate with each other in an online forum with weekly seminars. They are asked to share unpublished data. They are asked to share reagents. And they are asked to post all of their work on an archive before it gets published. And finally, we ask them all to publish in an open access format so that everyone of interest in the community has access to the work that we've funded. Well, here's a snapshot of where we stand now, 5 years later. We have 35 teams, 163 laboratories in 80 different institutions in 14 countries around the world. We've invested now over $500 million and likely more in the years ahead. The teams are gender balanced with many women in leading positions and a very large number of early career investigators. Every team had to have such an early career investigator. Finally, in my last slide, we do occasionally have very nice in-person meetings. Our first at the end of the pandemic was in the Bahamas. Not a bad place to have a meeting. Here I am standing next to Academia and to our team leaders from the Michael J. Fox Foundation and all of the principal investigators of all 163 labs arrayed up to the top. Well, this is a billion-dollar gamble. It's a gamble on the notion that an intractable problem like Parkinson's can be broken by a meaningful interaction among the best scientists in the world who are willing to share their results and not just hoard them to themselves. We feel that this model can be a very effective model for other intractable problems. And in this case, we have benefited enormously by the generosity of Mr. Sergey Brin. Thank you for your attention.
Randy, thank you so much for your truly moving and inspiring keynote. Due to a limited timing, we have to save your questions during the panel session. Now, I'd like to move our session into the panel discussion part. This session really is aiming to explore what are the cutting-edge research, at least some of them in life science, and how they are translated into the real-world applications that improve health and longevity. And we are privileged to have 4 distinguished panelists in addition to Dr. Schekman. And all these panelists are leaders in life science research with complementary expertise. Let's welcome them onto the stage. So let me first briefly introduce our panelists, and let me start with the ladies. Dr. Helen Blau is a world leader in stem cell aging research, with her research focused on muscle stem cells. And she is the Baxter Foundation Professor and the Director of Baxter Laboratory for Stem Cell Research at Stanford University. She is a member of the U.S. National Academy of Sciences, a member of the American Academy of Sciences, a National Academy of Medicine, and National Academy of Engineering. And she's recently also elected, by saying recently, last month, to the British Royal Society. And on top of all that, she's a member of the Pontifical Academy of Sciences. So let's welcome Dr. Helen Blau. And Dr. Jian Cui is a renowned oncology drug designer. She's a lead inventor of 3 FDA-approved oncology medicines and multiple clinical compounds. Dr. Cui is the co-founder, president, and CEO of Blossom Hill Therapeutics. Prior to that, Dr. Cui is the scientific founder of Turning Point Therapeutics. Dr. Cui was elected also to the National Academy of Engineering this year. Congratulations, and let's give Dr. Cui a hand. Now, Dr. Liqin Luo, sitting in the middle, he's a world leader in neuroscience, with his research focused on how neural circuitry are built during development and how these circuitry architectures will specify the function of neural activities in the adults. And he is the Anne and Bill Swindoll Professor at Stanford University and also an investigator of the Howard Hughes Medical Institute. Dr. Luo is a member of National Academy of Sciences and the American Academy of Arts and Science. Let's welcome him. Last but not least, Dr. Ziyang Zhang is a young star in using synthetic chemistry approach against human diseases. His lab creates chemical tools to tweak our immune system and enable new therapeutic mechanisms for cancer and autoimmune diseases. He is an assistant professor of chemistry at UC Berkeley. He has co-authored 30 research papers and is the inventor of 22 patents despite his young age. Let's welcome Dr. Yang. Okay, to kick off the discussion, I'd like to ask each panelist to say a few words about themselves that was not covered by me, and also their relevance to this discussion. Dr. Blau, could you start with?
Sure. Well, I was born in London, and I grew up in Europe. And I came to the US for graduate school on a scholarship to Harvard. And I thought I would just stay 1 year because I knew they gave you a master's degree after that, and then I could return to Europe. But I was so excited by the fact that it seemed like your energy and creativity were the limit in the US, and that was a contrast to Europe. So I ended up staying. And as you heard, I'm now a professor at Stanford University and the director of the Baxter Lab for Stem Cell Biology. And I started my career challenging a dogma, which was that specialized cells that function in your liver or in your skin are irreversibly determined for that fate. Once they are a liver cell, that's it. And I thought maybe that was not the case, and I devised a system for challenging that dogma and fused cells together, mouse muscle with human cells from all different lineages and specializations, and I showed that you could activate human muscle genes in these cell types that normally never would express them. So it overturned this dogma that differentiation was fixed and irreversible and showed that there's a lot of plasticity in the differentiated state. More recently, I've decided to approach a more audacious goal even, and that is to try and reverse aging, because I think that is a huge problem that we all face as we get older. And the reason I'm here is because Asian Americans have so profoundly impacted my life, my research, as colleagues and contributors and friends, such as Haifen, and it's been invaluable to my career. In addition to which, I'm a grandmother of 2 Asian Americans. I'm very proud of that.
Thank you. Now, in this audio sequence from left to right, Dr. Low, please.
Hi, I was born in Shanghai, and I was very lucky to get a position at Stanford 28 years ago. So I've been here since then. And in our lab, we study how nerve cells or neurons, what the image that you see from Randy's talk, make precise connections with their partners during development so that we can establish functional circuits that underlie our ability to sense, act, remember, and think. And also how these architectures enable these specific functions. We study these questions in model systems like mice and fruit flies because we can use precise manipulations to establish the causal link between genes, neurons, circuits, and animal behavior. And many of these discoveries should be generalizable to our human brain as well.
Great, thank you. Dr. Cui.
Hi, and thanks to the organizer, and have the great opportunity to come here. And so this is a really great event. And my name is Lincei, and I'm from China also, and it's a Ziyang, and I hope many of you visit that. That is a very historic city. And I graduated from University of Science and Technology of China, and major in chemistry, and then came to US, and that is a kind of 1988 to Ohio State University, and get my education and get my PhD degree in organic chemistry. So after that, and I did the very short 1-year postdoc, and then go to the pharmaceutical industry, biotech. So gradually evolved, and from organic chemist and to the medicinal chemist. So kind of evolving from organic chemist to the medicinal chemist actually is take a lot of effort. And because the training is effort, training is a chemist, and however is differently defined, it's a medicinal. So that means during the industry career, and learned quite a lot of the knowledge behind the disease, and also how actually to make the drug, and that is oral bioavailable, not toxic, and really perform perfectly and in human. So that is my whole industry. Still, now I'm still learning, and Haifeng, introduced me, and because now I'm very proudly and define myself as a drug designer. And so as a drug designer, and it's definitely and need to truly understand the protein structure, the dynamics, and then how actually position a drug and differently, perfectly, and into the protein, and then deliver to the patient. And my passion and is translational, and from the basic research and to the patient. So as the the drug designer, I feel it's a very special position, because all of any target and some huge effort from the community, from basic research and understand the mechanism, and it is the the drug delivered to the patient and then function there. And so I do feel this is the responsibility, duty actually, how to make the drug actually perfectly and function in human. And after I worked in the pharmaceutical industry, got the really training at Pfizer, and I resigned from Pfizer, and then started a turning point. So the reason started my own company, and that is after so many years of effort, and I definitely have my own insight how to pursue the drug discovery, how actually which, and I can pick the the target that is really immediately need and by patient. And so we work on the drug resistant at a turning point. And after, now it's kind of the drug that is back to 2013. And now the drug already approved by FDA. And I have my second journey, and this Blossom Hill Therapeutics. And we continue work on the drug resistant and also novel drug and associated with oncology and also autoimmune disease. Thank you.
Thank you, Dr. Cui. Now, Dr. Zhang, please.
Hi, thank you, Haifan. Thank you to the organizers, and really thank you to every single one of you sitting here. My name is Ziyang, and like many one of you, I came to the US to pursue grad school. I was a student in chemistry. I still think I am a student of chemistry. And, back then, I was learning how to make molecules, make things that never existed in the universe. And one thing I realized is, why are we making these molecules? And the goal is really to ask how these molecules can affect human health and maybe in a disease setting, or use them as tools to help us understand how things work. And as my study evolved, and I realized one thing: the same mechanisms and principles we use to synthesize chemical compounds are operating in the biological system, and sometimes are the reasons why diseases emerge. So I thought I could harness some of the chemical reactivity and chemical mechanisms and use it to design new therapeutic approaches. That's what I spent my postdoc research time on, and now I have an interest in our own immune system. It's fascinating to me why sometimes this powerful system just doesn't work the way it likes to. And I'm trying to design and maybe create chemical tools to help us understand how we can tweak our immune system in cancer and autoimmune diseases. So excited for the discussion.
Thank you, Dr. Zhang. Now let's dive into discussion. As you know that there are many, many fascinating questions in biology, and one of the most intriguing is how our brains got wired up. And Dr. Lu, as a leading neurobiologist, can you share with us a specific question that you're addressing in this area, and how your lab is tackling this question?
Yeah, thank you, Haifan. So the brain, brain wiring in general can be thought of as two different forces. One is how your genome encodes instruction to wire up your brain with molecules. These are called hard wiring. And the second component is experience-dependent modification, how you sculpt the hard wiring component. Actually, in many circuits, the hard wiring gets a lot of things done. How so? So there was a hypothesis developed 60 years ago that each neuron make molecules that are put on their surface so they can distinguish one from the other, and then so they can find precise partners. And indeed, we call these wiring molecules, and indeed many wiring molecules have been identified over the last decades. But the hypothesis also introduced a very interesting problem. How, our human brain has 100 billion neurons making 100 trillion connections, but our human genome has only 20,000 genes, only a fraction of them, at most 2,000, at most, wiring molecules. How can you use a limited number of molecules to wire up so many connections in a so precise manner? So one solution often posed is combinatorial coding. For example, the on-off state of 6 molecules can give you 2 to the 6th or 64 different combinations. But these are largely theoretical and has not been experimentally tested. Recently, because of technology innovations, some our colleagues and I in the lab recently developed in a fruit fly olfactory system to allow animals to smell, that we can actually change the expression of 5 molecules in one particular type of sensory neurons that are normally used to sense the pheromones that inhibit male to mate with another male. And we can channel it precisely to send the information to the target neurons that are normally used to promote mating. And we can do such a precise switch so that the resulting transformed flies, actually males will chase after other males. So this is an interesting behavioral results, but it's really showcased that our understanding that we can manipulate molecules in a very precise way to alter the complex wiring problem.
Right. Thank you, Dr. Liu. You really, thank you for a very lucid explanation of such a complex process. And we know that brain signaling really boiled down to molecular level is molecular movement within and between cells. And Dr. Schekman, as an expert in this area of study, can you share with us your study in this area and how your basic research can impact on actually bedside and the real world situation?
Yes, thank you, Helen. So for many years, my lab has studied how proteins get manufactured inside of a cell and exported outside of the cell. The cells in our body have all of the genes housed inside of the nucleus inside the cell, roughly 23,000 genes, at least 23,000 different protein molecules. All of them are made inside the cell. But some of these molecules have to escape, and the escape has to be somewhat selective. Hormones, molecules like insulin, neurotransmitters have to get out of the cell to communicate between cells. And the way this happens is by little particles that convey things to and fro. They're called membrane vesicles. All the cells in our body make little membrane vesicles that encapsulate proteins that are secreted outside of the cell. So the contour of this process was known by the 1960s, early 1970s. But when I started my career at Berkeley, nothing was known about how this process is operated at a molecular level. What are the mechanisms that allow some molecules to be recognized, to be encapsulated, to be moved, and then to be discharged outside of a cell? So for much of my career, I studied this process in an organism that hadn't really been used to study this, but it was perfectly suited. It's called baker's yeast. You know baker's yeast, how useful it is commercially. But it turns out, as an experimental organism, it's also profoundly useful. There are simple ways of doing genetics, isolating mutants, simple ways of doing biochemistry. You can grow huge quantities cheaply. They're all uniform. The cells are all uniform. So my lab discovered the genes that are required for protein secretion. And when it became possible to sequence these genes from yeast, and subsequently when the human genome was sequenced, it became apparent that these genes are evolutionarily conserved. That is, the processes that we use for our nerve cells to communicate with each other are processes that that evolved 2 billion years ago, some of them even 3 billion years ago on Earth, and the bits and pieces that have been passed down over all those years to be preserved in structure and function. So that knowledge of evolutionary conservation allowed the biotech industry in the early 1980s to use yeast as a platform for the manufacture of clinically important proteins. And so I helped a local biotech company engineer the gene for human insulin into yeast, and it became possible to trick yeast cells into manufacturing and exporting human insulin, a protein that it's never seen. And now, 1/3 of the world's supply of human recombinant insulin is made in giant fermentation vats, producing metric tons of insulin for treatment of diabetics. In more recent years, my lab has shifted away from yeast to look at things that are of greater consequence for human genetic diseases and physiology. And we've been focused on a kind of a vesicle that isn't restricted to the cell. In fact, some vesicles that are manufactured inside of cells are themselves exported outside of the cell. So in our bloodstream, in our saliva and urine, all of our bodily fluids, there are billions of these little vesicles floating around that can be sampled in a liquid biopsy. And these are vesicles that were manufactured by one or another tissue in the body, so they become kind of sentinels that express the physiologic state of the cell from which they were emitted. And so by developing very simple, powerful tools, biotech companies have found ways to recognize a small number of vesicles that may predict the advancement of disease. And so there are now diagnostic tools being developed to recognize very early stages of cancer that would not otherwise be diagnosed by any other means. And so this is going to be an emerging, very powerful technology that will be based simply on liquid biopsy. So I'm very excited about that, and my research continues at a very basic level to understand how these vesicles are made, how they're exported, and what their purpose may be.
Thank you very much, Randy. And Randy's example indeed illustrates how fundamental research can lead to such profound applications in medicine and in industry. So now I'd like to switch the topic a bit to yet another very fascinating question in biology, the one that perhaps all of us care about the most, namely aging. So Dr. Blau, as an expert in studying stem cell aging, especially in the muscle system, can you advise us on how we can combat the loss of muscle stem cells and the muscle itself as a mass that's so debilitating to people when they get old?
So thank you, Ivan. Yes, you've heard a lot about how we're losing our brain functions. Well, equally important, I think, is brawn. And brain and brawn there's an interplay. We care about our muscles. And there is, as you know, with aging, you lose muscle function. You lose 10% of your muscle mass and strength per decade after the age of 50. And it's very hard just to maintain it, almost impossible to build it after a certain age. And that muscle wasting can lead to major dysfunction and inability to do daily tasks of life, like walking. And it leads to an increased incidence of falls, and then institutionalization and loss of independence, and then also early death. So there's a major unmet need for drugs that can combat that. There are no drugs for building muscle. And you probably know people, even younger people, who were bedridden due to COVID or because they broke a hip. And during that time of 2 or 3 weeks in bed, their muscles atrophy, they shrink and they lose their strength. And I get these heart-wrenching emails from people who say, I used to be so active and now I can't go up the stairs and I have to go to assisted living. So this has motivated me to direct my attention to this great need. And so we screened for drugs that could activate muscle stem cells and build muscle strength. And we found that there's a metabolite that is in your bodies, in your muscles. It's part of your natural healing mechanism. It's the metabolite prostaglandin E2. And the levels of this metabolite are very tightly controlled, and they are essential to your muscle stem cell function. If you don't have the receptor on the muscle stem cells, they can't respond to PGE2, they can't repair a damage. And equally important, if you give a person an NSAID, something like ibuprofen, you block the synthesis of your own prostaglandin E2. So it's essential to muscle stem cell function. And then we found that this prostaglandin declines with aging. And that is due to an increase in the degrading enzyme, the prostaglandin degrading enzyme, PGDH, which we've called a gerozyme, an aging-associated enzyme. And it just goes up a little bit, maybe twofold, but that has a dramatic effect on the muscles. If we overexpress this enzyme in young muscles, they shrink and weaken within 1 month in mice. And if we block the activity of this enzyme in aged muscles, we can increase strength by 15% in 1 month. So these findings, I think, are very exciting, and my goal is to translate that and try to get it to people. And so I founded a company that is doing that and developing drugs for human use that are oral to build muscle with aging and disease.
Thank you, Helen. Let's all follow Dr. Blau's advice to heart and hope one day we can benefit directly from her discovery of gerozymes. As you all know, that research in biology, just like in any other disciplines, requires a lot of interdisciplinary skills. And Ziyang here is one great example. He was trained as a chemist, but now really working on drug designs that's targeting cancer signaling and immunology. So Ziyang, do you feel being a person, so-called outsider, walking from another field into a complex life science field, is this a challenge or is an opportunity, or if both? And if so, what's your personal perspective on this?
Yeah, thank you, Haifan. This is a great question. So, you know, when I was a trainee, and the way we're trained to think is that when you have a problem, you need the right tools to solve it. But what's not told to me, at least, is sometimes the best tool is not the ones you're familiar with, or it's not the one that everybody says you should use. So, you know, this is the case many times in life science already. I think the field as a whole has benefited a lot from physics, from chemistry. Microscopy is a good example. What I realize is sometimes you just need a totally different mindset, and this idea has been mentioned many times today. So I'll give you one example of my own. So when I was working as a postdoc, I was looking at this famous oncogene called KRAS G12D that's in about 10 to 15% of all cancer patients. We were trying to find a chemical strategy to capture this mutant, but not the normal version, so that the patient's normal body can still function. So we started looking into a very particular chemotype called the beta-lactones, because based on our chemical understanding, we thought that has a chance. And we did a lot of work, and we spent a lot of time understanding the reactivity. And eventually, we thought we had a pretty good idea, a new system. But that's when we started to read more papers and realized that we did not come up with any new technology. The same thing has been used in the polymer industry for 30 years, dating back to the 1970s. So in this sense, if we think backwards, if we had we known the chemical principles in polymer science and integrated that early into the project, we would have figured out the solution earlier. So this is really a personal story that made me realize sometimes you just have to look outside your own field. And I think I have personally benefited from this by keeping an open mind, and I know a lot of you share the same opinion. So I think I would just conclude by saying I feel that being willing to accept new knowledge, even if it's not immediately relevant to your research, is like learning a new language. For us, we speak multiple languages. I think all of us do. And nobody can say learning a new language is immediately useful. You can't say, I'm going to use it for this right away, unless you're traveling to a new country. But it's going to pay off at some point, and it's going to become handy. It's going to make you, enable you to make new connections with new people. And I think the same with, new ideas and new techniques in research as well. My answer.
Thank you, Ziyang. Your comments are very inspiring to me, and I hope especially they are very inspiring to young scientists, students, and postdocs in the audience. Now, due to time limitation, I will save my second or third round of questions for you guys. And I think we should open the floor for the audience. So please feel free to raise your hand if you have any questions for Dr. Schekman's talk or any of panelists. Number 3 row, please. Yeah.
I have a question. So like for biology, do you guys use like software to simulate it, or do you guys like experiment on it?
Maybe Liquin, you'll start with this. Can you repeat your question, please?
So like for biology, do you simulate it, or do you experiment on it, or both?
Both.
Yeah.
Both? Okay.
Yeah, traditionally, experiment we are prioritized, but right now, because of the so many computational tools and also powers, I would say in the neuroscience at least, computational neuroscience becomes more and more prevalent.
Oh, okay. Thank you.
Yeah. You can formulate, I think, more precise hypotheses and using the data available, and then you test it to know whether for sure it is working.
Any other panelists want to add to these comments? How about in the audience? Other questions?
Yeah, hi.
The one gentleman in the middle here, and then the one after you.
Yeah, I have a question on this wet lab and the AI. So I'm doing the AI for life science. So according to my experience, like wet lab scientists, they do not accept AI's suggestion. But my experience telling that AI doesn't work until we actually change the way to do the experiment. So how do you guys think about this kind of intrusion kind of way to interact directly with the wet lab and the experimental design? Because this, that's my feeling, because otherwise it's like I'm doing a cancer vaccine, it's so hard. AI think we already done it, but it's not. But how can we actually interact with the real experiment? That's my kind of question. I'm asking for your suggestions. How can we, from AI's side, to help or to learn the experiment and the insights? Because you guys have lots of insights, but we want to encapsulate or abstract into AI algorithms.
Well, I can speak to that. With the gerrzyme that comes up with aging, one of the big questions is what turns it on? Why does it become more expressed with aging? And so we've collaborated with an AI expert, a bioinformatics expert at Stanford, Anshul Kundaji. And we use methods in the lab to reveal the DNA and what's accessible. But what he can do with AI is see motifs, binding sites that we would not be able to see with our methods. And he can develop algorithms and predict how different proteins are binding to turn on the gerrzyme. And it's a fantastic tool, and then we can test it in the laboratory. So I think that AI is informing what we do and making our hypotheses more precise.
Okay.
Let me correct a misimpression that you may have created that AI has not been successful. In fact, it's been enormously successful. One of the great triumphs just in the past 5 years of AI has been a program called AlphaFold, a DeepMind, relying on the enormous genome database available. They were able to develop an algorithm that predicts quite accurately the folded structure of some 250 million predicted proteins that were deposited online just 2 years ago, to a level of precision that had not yet been possible by any previous algorithm. That was intimately dependent on this enormous data set and on the programs at DeepMind, the AlphaFold in particular.
That's great. Both Helen and Randy's answers are all illustrating how important AI is to biology. But I think Dr. Liqin Lu, being a neuroscientist with expertise in neurowiring and networking, could you provide some counter perspective in a reverse way, actually how neuroscience research might have helped the AI?
Yeah. So you're talking about, there is a deep relationship between artificial intelligence and the real intelligence, which is the brain. And in fact, some of the engines that are behind recent revolution of AI comes from the architecture of the deep neural network, which was inspired by the mammalian visual cortex, discovered decades ago. And of course, the detailed implementation of how these works between deep neural network and the cortex are quite different. But nevertheless, understanding the neuroscience architecture can inspire AI. On the other hand, we really still are far away from understanding how information is processed in the brain, and we can benefit a lot from AI, just like hopefully someday, like the protein structure can be solved by largely the AI-based deep learning network. On the other hand, actually, in the real brain, there are many different architectures of the circuits that seamlessly work together. We still don't know how they work together, but if we one day figure it out, maybe we can implement these back to AI and to provide some possibilities to generate artificial general intelligence that people have been chasing after.
Right. So we are running a bit late, so I suggest we have 2 more questions, quick questions and succinct answers. That gentleman and this young lady, you've been raising hand for a while, near the aisle in the central region. Right. Yes. Yeah.
Thank you all for coming. I have a simple question. As people often saying, aging is never age. Just wonder, do we have any scientific support behind this? Thank you.
I've never heard that one before. But if so, I'd love to understand what the key is. No, I can't speak to that. Can one of you?
Any other panelists want to comment on that philosophical topic?
Wishful thinking.
All right. Now, please.
Yeah, so I have a question for Dr. Luo and also Dr. Blau. You've talked about, I'm a physician and a scientist, so I've seen, the aging, aging research on aging has been invested a lot, especially on brain function. So, but I'm seeing is in the brain early development from infant, from fetus, from infant. I was wondering, is there any more advanced research as well? But I've seen is, I don't see enough investment in this area, in the early development, neurodevelopmental research. Instead of, when we're getting older, the last end of the stage of life, we want higher quality of life, for the brain function degrade, aging. And on the other side is early developmental, neuro development from a physician standpoint. It also needs a lot of research. And I was wondering, is there any, more, and AI can help us well. I was on an update on this.
Certainly the neurodevelopment, we still have a lot to learn. We know a little bit more, about the embryonic development, there was a big focus. But for example, once we are born to adolescence, the early childhood, this period, I would say, it's very little is known because of, the technology limitations, I would say, in the last decades. But now, this is a very critical period, not only for us to have a healthy life, but also a lot of the disease. In fact, even though their manifestation is in your, like a lot of neuropsychiatric disorders like schizophrenia, bipolar, even though the first symptom comes when you're teens or in the 20s, but a lot of the seeds were set up in the early development. And even contributing to some of the aging. Yeah. Some of the seeds could be very early. So we really should invest more time, effort to study these kind of development. Maybe Helen can add more.
With respect to the muscle, we're studying, we have access to these biopsies, muscle biopsies, from people at different stages. And in particular, it's fascinating that we have samples from people who are healthy aged, and age-matched with those who have sarcopenia, which is profound muscle wasting. So we want to understand the gene signature, what makes a person frail versus healthy, and can you predict ahead of time? And what kinds of things can you do to intervene in those pathways? And we're using AI to understand these gene signatures and to understand the gene expression and the pathways that go awry. And one of them is the gerozyme. And we're very interested in how that might rescue. But I think prediction is very important in understanding the development and progression of aging and disease.
Great. I have a comment is...
sorry, we have to cut this off. Please save your great questions to approach panelists during the lunch break. However, before concluding the session, I do want to give Dr. Jiang Cui the final words by answering one of my many questions. I always admire those people, the courageous scientists, who can take a deep jump from research to biotech industry to benefit the society directly from what they discovered. And Dr. Cui is a great example of that. So, Jiang, can you share with us your perspective, how difficult, how exciting, how challenging it is for you to move from a bench scientist on the fundamental research side all the way now become a CEO in a biotech company?
Yeah, it's kind of a very different experience and working in the industry compared to academic. And however, science is kind of we share the science and we kind of move science and from the different angle. And so everybody know and kind of a drug and getting more and more expensive. One of the reasons is kind of the failing and in the clinical trial, it doesn't work. And so kind of working in the industry, and one of the fundamental and the question is, kind of when we start any project, and what is the target and related to the disease. And so all of the failing, that means we definitely have a lot of basic research hypothesis there. However, is that true in the human? And so this is kind of, and also, and in the translational part, and is one of the major things is missing is the model. And we definitely cannot, mouse model may not predict what is a human. So that is kind of a major block there. And so kind of from industry point, we always come from patient and what is really need there. And so that is also the passion, why we work hard and how to solve the patient's problem. And then we always kind of do the research to see what kind of knowledge about this disease and around there. And, kind of, and which one actually is more mature and to move to the transfer to the clinic. And it used, kind of take, multiple decades and to validate one target and then to the human. And it's a lot of basic research around. And however, with the technology advanced and the next-gen sequence, and we understand more fundamental of genomics and also the mutation, and from the big data base, we know and relationship to the disease. And then this process can be very fast. And I just give you the personal experience. And from the 2007, and discovered EML for ALK and fusion in non-small cell lung cancer. So even so, it's only 5 to 7%, and a non-small cell lung cancer patient have this disease, this fusion gene. And so kind of it is a Pfizer, we have ALK inhibitor there, and tested in human, and within, it's probably half a year. That's a Nature publication is in August, and then for the first patient in December, Christmas time. And so that is kind of the patient responds very well, and then the whole facilitates the whole clinical trial. And then from the 2011, the approved the drug, and this is unprecedented. From identify this fusion gene and to the drug approval, only 4 years. And so that is a demonstrate, kind of the different speed and how the, close relationship from industry to the academic. And also kind of it is not easy to identify the validated target. And so this is kind of when we first work on the validated target, and we probably in the rush and to get the molecule, work in the clinic, and validate this is the disease-driven target there. And we probably got the PFS, kind of for this I gave the example, and crizotinib, and it's a PFS progression-free surviving time, and it's about 10.9 months. And so, and then develop resistant. So that is the kind of the mission for next one, and how to solve this, resistant, and then make the, progression-free surviving time is longer. So in the past, ASCO meeting, and finally we read out the third generation lorlatinib. That is another molecule I work on, And so, it's hard to imagine as the small molecule achieve the progression-free surviving time more than 60 months, 60 months, more than 5 years for non-small cell lung cancer, and still, 60% patient on this drug. So that is kind of from the result and validate how we should approach and for the drug development. And actually, that is the kind of back first from the basic research continue to improve and how we design the drug and approach how we approach the problem. Thank you.
Thank you, Jen. Let's thank all panelists for sharing with us their words of wisdom. And I want to thank all of you for your very engaging discussion. This concludes the session, and hope you enjoy the lunch and the rest of the program. Thank you.